Hi, my name is Meghana Karamuri, and today I will be presenting about codon processing to create a model that predicts translation efficacy. To solve this problem, what I did was create a Python tool called Codon Count. Before I explain my Codon Count Python tool, I would like to first provide a little bit of background information. Different codons bind differently to amino acids. Based on the nature of the codon triplet pairs, some codon triplets bind more effectively to amino acids than others. What we wanted to do was create a tool that will help us see if translational differences exist within codons. Another way to look at the optimality of codons to see if there are translational differences is to examine the start site of an RNA sequence. Specifically, the codon count tool examines the Kozak sequence, which is 6 nucleotides before the start codon and 1 after the start codon. This is a 10 nucleotide sequence. Based on how the sequence is built, certain start sites are more optimal or stronger. To build a program to process codons, I used the BioPython tool to fetch and access sequences from the NCBI database and analyze them in Python. To explain this project, I will first start with the project workflow. We started with isoform level data, which have ENST ensemble IDs. Using these IDs, we use the NCBI database or biodb.net, which are tools which can be used in order to retrieve the refseq IDs. These refseq IDs could be used to then query the NCBI database and retrieve GenBank files containing sequence information. This is a sample shortened GenBank file for the gene with the refseq ID NM001100.4. Over here, you can see basic features, information about the genome sequence. After obtaining sequence information from these GenBank files, the codon processing code, or the tool that I built, calculates the codon frequencies, extracts the Kozak sequence, and encodes the sequence into a 64-char representation for later use. This program basically outputs 2 CSV files. One of these files has codon frequency data and the Kozak sequence data, and another file has the 64-character encoded sequence. Here you can see a sample output of the codon frequency data, which basically takes in a sequence and calculates how many times the specific codon appears in the sequence. The output file is shown above. This is a sample output of the Kozak sequence data, which is the 10 nucleotides around the start site. It can be used to predict translation efficacy for a certain mRNA sequence. This is an example of the 64-character encoding. Basically, each codon is encoded by a specific sequence or character which represents a codon triplet. The codon count tool then produces an output using the 64-character encoded sequence and shows basically the sequence in an encoded representation. This basically takes up less space and data and can be used for easy analysis.